first of all, I'm very excited about this seminar for two reasons. First of all, it's going to be reporting on a very, very interesting study, one of these national longitudinal studies, which are now, thanks to David and Blair, are being analyzed, as I understand it, from a multi-level cultural evolutionary perspective. So isn't that awesome? And secondly, because David is my academic grandchild. He is the PhD of one of my PhDs, Rick O'Gorman. And Blair, you are the PhD of Jim Cohen.
Yes, he's on my dissertation committee, correct, yes.
And Dennis Prophet.
Yes.
And so this is like a younger generation. And isn't that awesome? So I think with that, why don't we just head right in and hear what you have to say?
Yeah, absolutely. David, I'm not sure who's presenting. If you all could do like a presenter view, that would be great.
Yeah. So I'm not sure how I can set up a presenter view for you from here. So you seeing slides?
Just present it. I have my notes up. It'll be fine.
Yeah. Okay.
Oh, the wonders of technology until it doesn't work. So anyway, so thank you all for coming to the seminar. And so David and I are going to share this talk together. And I was going to begin by sort of defining and giving an overview of social baseline theory. And then I think the plan is for David to take over and discuss more of the UK Household Longitudinal Study and then the research plans that are sort of arising from this collaboration. So David, you can go to the next slide. So humans are inherently social creatures. And in fact, our social environment and social support is so vital to our functioning that the more socially isolated that we are, the more likely we are to die. And this isn't hyperbole. This is actually, and you can go ahead, David, this is actually well supported with research. So in a study published in 2010, Holt-Lunstad conducted a meta-analysis to determine the extent to which social relationships predict our mortality, so our likelihood to die. And they found that social isolation was as big of a risk factor as smoking. You want to hit the two, there you go. And an even larger risk factor for mortality than physical activity, body mass index, and high blood pressure. So what you can see here are the effect sizes of each of these. And social relationships, again, is as big of a factor as smoking for mortality, and even bigger than things that we typically think of as very harmful for us. And so the social environment is incredibly important to all of us. So you want to go ahead. So social baseline theory recognizes the fundamental importance of our social environment. And it is strikingly different from traditional approaches to studying our mind and behavior, which typically focus on studying cognitive processes in isolation. So I really love this picture because it kind of illustrates for me what social baseline theory is. And so at its core, to define it, social baseline theory suggests that the human brain assumes access to social relationships. So in other words, it assumes the presence of trusted social connections. And importantly, a lack of access to social resources, such as living in isolation, can increase the cognitive and physiological burden in individuals. The reason why I like this picture so much is because social baseline theory is a perspective, and I think this illustrates this, that unifies research in behavioral ecology, in perception science, and cognitive neuroscience, all of that in addition to really emphasizing an individual differences approach. So what my plan is I'm going to take a few moments to talk about each of these different areas and how they contribute to social baseline theory in turn. Okay, David. So I'm going to begin with behavioral ecology. So one of the first things that we should recognize is that social baseline theory relies on an economy of action framework. And what this means is that for survival, we must consume more energy than we expend. And so within this framework, this means that humans and animals will behave in a manner that maximizes our energetic gains and minimizes our energetic costs. And the field of behavioral ecology is rife with examples of balancing our energetic intake versus expenditure. So, for example, honeybees will maximize their energy efficiency with partial crop loading. And so to explain what that might mean, you can kind of liken this to carrying groceries home from the store. If you have a very long walk from your house to the grocery store, you would typically buy less groceries to carry, right? So maybe make more trips, but carry less groceries. But if you had a short walk back to your house, you might sort of load up on your groceries in all in one trip. And so an economy of action framework would suggest that, or would predict that, to maximize the net energy gain, the farther away the bees have to fly to get to the nectar, the lower the load they should carry. And this is in fact what they do. And so what we find is lots of evidence in the animal kingdom that this is what we are doing. We're maximizing our gains and minimizing our energetic costs. Now, take another example from behavioral ecology, which are ostriches. If you enclose ostriches in a pen with a limited amount of food, so if you're studying ostriches and you put them in a pen with a limited amount of food, they actually consume more food when they're in a group than when they're alone. And this seems really counterintuitive because if there are more ostriches in the pen, they should have to share the food with each other. But what happens is that ostriches actually have to put their head down to eat, and that makes them vulnerable to predators. So if ostriches are eating alone, what happens is they have to stop every so often to scan the environment to check and look for predators. But in a group, they can share the cost of being vigilant for these predators. And so when they're in a group individually, they are able to actually consume more food because they're sharing the cost of watching for predators. And so that ends up being a net gain for them. And so social baseline theory posits that the presence of other people, the presence of others in our social environment, it serves as an indicator for more available resources for ourselves. And this is because others can share the cognitive and the metabolic load of acting in the environment. So the presence of other people can signal to us that we have more resources available because we're sharing the cost of acting. So David, if you want to go forward. Now, this is very closely related to the free energy principle. And so the free energy principle, or another, sometimes it's often referred to as the Bayesian brain, was proposed by Friston in 2010. So this suggests that our brain actually optimizes behavior by minimizing the amount of prediction error or surprise in the environment. So I really like this graphic because it's a good demonstration of what we mean by minimizing surprise. So from this perspective, stable social environments represents a way for a brain to reduce its free energy burden. So to give an example of this, if a child has a stable, loving caregiver that consistently provides them with food and shelter, there's not a lot of surprise in their day-to-day lives. And they have a very low free energy budget or free energy burden. But if you compare that to someone who has an unstable caregiver, there is a lot of surprise, a lot of variability. And so they would have a very high free energy burden. And so in this case, a stable social environment or stable amounts of social support represents a lower cost of acting in the environment because there's less of an unpredictability in the environment. So, David, go ahead. Now, we also find evidence for social baseline theory in visual perception. And so we know that our perceptual system consistently overestimates the slants of hills. Hills appear much steeper than they actually are. So for example, for this picture, when asked to report how steep Lombard Street is, and this is in San Francisco, if you ask participants to report how steep it is in degrees from 0 to 90, where 0 is perfectly flat and 90 is a vertical wall, most people will report it appears around 60 degrees steep. There's a variation, but typically very steep. But in fact, this street is 18 degrees steep. By law, that's the steepest road, paved road in San Francisco. And in fact, we are not able to walk down a street that is more than 25 degrees steep biomechanically without breaking into a run. And so we have to ask ourselves what's happening here. You can go ahead, David. And so what we believe is happening, that for physical resources, the cost of acting in the environment, and you can go ahead and pull this out if you want, the cost of acting in the environment is calculated as the energy required to perform the action relative to the amount of physiological resources. So hills are bioenergetically more costly to climb than flat ground. And so our perceptual system is exaggerating the steepness of the hill to inform us about the physiological cost of climbing it. So within this account, if you either increase the bioenergetic cost of the action itself or decrease the amount of physiological resources, the net cost of acting in the environment is higher. And so hills should appear steeper and distances should appear further away. Go ahead. And this is exactly what we find. So if we are less physically fit, wearing a heavy backpack, elderly, or even fatigued, hills will appear steeper and distances will appear further away. Now, one of the strongest lines of evidence for this account comes from studies in individual differences. And so Jonathan Zadra in 2016 published a study that found that individual differences in how physically fit an individual was actually predicted how much that individual overestimated distance, both before and after exercising. So according to social baseline theory, if we think about this, other individuals represent more resources in the environment. And so if this is the case, then friends should make hills appear less steep because friends are representing other resources. Go ahead, David. And this is also exactly what we find in the research. And so this was a study published by Simone Schnall in 2008. And what we have on here are two different graphs. I'll just explain the graphs to you quickly. And sorry, I don't have a pointer, I can't point. But on the x-axis of each of the graphs is our verbal, visual, and haptic measurements of the hill. And so a verbal measurement is what I've already explained to you. You ask a person, how steep is the hill? How steep is the hill in degrees from 0 to 90? The visual measurement, you take a disc and you match the steepness of a hill to the steepness of a disc. And for a haptic measurement, you put your palm of your hand on a board and you measure the estimate of the hill with the palm of your hand and measure it that way. And so what I want to point out is that for both, so if in the graph on the left, what was happening is that participants were walking either with a friend or by themselves in the environment. If they're walking with a friend, they estimated the level of the steepness of the hills to be less steep than if they're walking alone. And this actually replicated in a second study where they didn't even have to actually be in the presence of their friend. If they just imagined a supportive friend for 5 minutes, the hill appeared less steep. And so in this way, social support is functioning as a physiological resource. And friends literally change the way we see the world around us. So if you want to go ahead to the next one. Now, while social baseline theory is interdisciplinary, the impetus for the idea actually began in cognitive neuroscience with a hand-holding study. And this was conducted and published by Jim Cohen, who David mentioned earlier, and his colleagues. So in his work, Jim was investigating the neural mechanisms behind the social regulation of threat. So in other words, social support and handholding helps regulate our emotional responses to threatening stimuli, for example, an electric shock. And so Jim was investigating what areas of the brain are associated with this regulation. And so in this study, he brought in married women and put them in an fMRI scanner. And he put the women under threat of a mild electric shock while they were either alone or holding their hands with a stranger or holding their hands with their spouses. Go ahead, David. And what he found is that the areas of the brain that typically respond to threat were more active when the individual was alone, less active when holding a stranger's hand, and even less active when holding their spouse's hand. Now, this is really surprising. So from a traditional standpoint, you can go back a little bit. So from a traditional standpoint in cognitive neuroscience, this wouldn't make sense. So typically when you add a cognitive process, so let's just give an example. Say you were asked to pay attention to 2 stimuli compared to 1. You would increase the amount of activity in the brain. So in this case, if you were trying to pay attention to your partner hand holding and this possibility that I'm going to receive an electric shock, what you would expect is that you should have seen more activity in the hand holding condition because you're adding cognitive processes. And what he found was the exact opposite. And so the idea is that this traditional perspective assumes that the individual is the default level of analysis, but instead our social baseline theory suggests that the default for humans is to expect a baseline of social support. And that others not only represent added resources in the environment, but also reduce our cognitive burdens of acting in the environment. And so in this case, literally, this was the basis for social baseline theory, that we assume social support. And in studying the individual alone, we're studying them at a cost. Now you can kind of take it. Now, interestingly, Cohen also found that the quality of the relationship between the spouses predicted how well the hand-holding attenuated the threat response, but only for their spouses. And you can click this circle here. So what you'll see is that in this middle, in this graph right here, in this middle column here, what you see is this wonderful, these negative correlations, which the higher the relationship or the better the quality of the relationship with the spouses, the more that attenuated the threat with hand-holding. And this, to me, brings up a really important point in social baseline theory, one that I've tried to emphasize, which is that we also must consider individual differences in our social baseline. So you can go ahead. So while we propose that social environments are inherently a part of our cognitive processes, it's also the case that individuals vary from each other in how integrated we are in our social environments. And that our social baselines are dynamic and can fluctuate with experiences. So you can first of all think about how individuals might vary from each other. So you might have some individuals who are, let's say, more extroverted than others or more introverted than others. But also, even within an individual, our social baselines are going to vary with our own experiences. So negative experiences would alter the social baseline, so the social environment may be less incorporated into our baselines, whereas positive experiences will result in our social environments overlapping more with our baselines. So this is a very important part of the theory, is to recognize that that while we do have social baselines, this is not a static process. You can go ahead. And so finally, I'd like to mention that social baseline theory does not strictly apply to individuals, but can be expanded to a hierarchical group level analysis as well. So for example, allostatic load is defined as the wear and tear on our physiological systems that comes from chronic stress. And so our social relationships can also experience a social allostatic load, where internal and external stressors can increase the regulatory load in the group, and that can erode the group's functioning over time. And again, how well the group as a dynamic force is functioning within our social baseline. Okay, next. Okay, so now that I've introduced some of the terms and the constructs and the evidence for social baseline theory, I'm going to turn this over to David, who will introduce the UK Household Longitudinal Study. and more of the connections and collaborations that we are considering.
Thanks, Blair. I'll just open. Right, is that presenting for everybody?
Yep, it is presenting.
Yep.
That's great.
My title slide, David? Oh, no, there you are, yeah.
Yeah. I say, to begin, I'd like to draw down a little bit deeper into some of the social baseline theory that Blair has just introduced us to. And following that, I'd like to introduce the UK HLS. Hereafter, I'll refer to it as Understanding Society. Then we'll revisit these motivating studies to identify research questions that we can explore using the Understanding Society data. So I first contacted Blair, having read her excellent review and extension paper. I was particularly interested in a better understanding of the environmental influences of social baselines and exploring the dynamic effects, social interactions, as my own research, which we may walk through a little later, is focused on understanding how the physical environment gets under the skin to affect our health and well-being. So something Blair didn't mention, but during her presentation is, although she did allude to the group level hierarchies or the group level hierarchies in the social allostatic framework, was the recent integration of social baseline theory with multi-level selection theory. So David and Jim Cohen have, in their paper, Groups as Organisms, published in the Clinical Psychology Review, may be subtitled Implications for Therapy and Training, but its ramifications of its main argument, namely that groups at multiple scales can be valid units of analysis, as they foreground in the final line, of this abstract, has implications for improving well-being at all scales, from individuals to the planet. So, the construct that Blair did closed her presentation on, the construct of social allostasis, through the work of Saxby and colleagues, begins, in our view, to ground both the behavioral and the physiological processes that David and Jim's insights regarding groups at multiple scales functioning as organisms in their own right. So when these three papers were published, I was doing my graduate training on the SOC-B program at the University of Essex, and I was a member of Meena Kumari's health research group. And during my time at Essex, and in an early meeting of a working group focused on composing a standardized approach to computing individual level allostatic load measures for understanding society, I introduced this social baseline theory, the multi-level selection theory, and the social allostasis construct to the conversation. And that started this ball rolling. So what is the Understanding Society data asset? So Understanding Society is the single largest investment in the UK's Economic and Social Research Council, recently securing a further £100 million in funding for the next 6 waves of data collection. It is a large-scale longitudinal household panel survey that has been running since 2009, collecting information annually from members of around 40,000 households across the UK. The study covers a wide range of topics, including income, employment, health, education, household composition, social lives, and other topics. By following the same households and individuals over time, it allows researchers to study how people's lives change and what factors influence these trajectories. It is considered a valuable resource in research across disciplines like economics, sociology, epidemiology, and public policy. Now, the topic that I was involved in during my training was the biomarker genetics and epigenetics. So the SOC-B doctoral training program center that I was working on aims to create a cadre of interdisciplinary researchers across the social and biological sciences. And Meena, Professor Meena Kumari, who is a biological and social epidemiology, is both the director of the center and an associate director of Understanding Society. So the SOC-B basically stands for biosocial, slightly the wrong way around, social biological, so biosocial research, which examines the interplay of social, behavioral, and biological factors, and data assets like understanding society, which combine these rich social and biological data sets are vital resources in this work. So what are the biological data assets that we have? And I saw Anna was on this call earlier. If she's still here, I want to thank Anna and give credit to this slide from one of her presentations. So along with all of what we might consider those conventional social data modules that are in longitudinal studies, understanding society also collected a decade or so ago bloods from participants, and we can now use the data that's been generated from analyzing those bloods in different ways. So we have this rich individual, household and area level longitudinal data and these biological data assets. And historically, the logic of the unit of analysis for this these data sets has been of the individual. And the proposal that we're putting together is to extend that to include groups at different scales. Now, the next few slides, particularly Anna helped me with quite some time ago. So if we just look at the top of these three R outputs, I'm just going to give you an account of what these numbers mean. So what we have is the biomarker information. So they're the sorts of data that we get from the blood that was taken, in this instance, blood from the participants. And for instance, the biomarker that I've used is C-reactive protein, which is a good measure of inflammation. We have data on approximately 13,000 people, but it breaks down for our purposes, quite interestingly, regarding the size of households. So if what we're interested in is looking at households as units of analysis, when we look at the number of participants that we have data for, where we have them from a single individual in a household, you can see that that's 4,943. So it would be difficult to run an analysis based on our ambition to look at the social baseline of that individual with that group. But as we move further to the right, as you can see, we have in the different households where there's 2 participants, where we have the biomarkers, we've got 2,139, and that's households, so that's double that number of participants. In the households with 3 participants, we have 192 instances of that. So you triple that number of participants because there are 3 participants in each of those houses. And then again with 28 for the number where we have 4 participants in a given household.
David, could I stop you there for a minute just for some clarifying questions?
Yes, of course. Go ahead, David.
Is that like 2 adults or?
It will be 2 adults minimum, because we didn't collect, or when the data was collected, the nurses did not collect bloods from anybody under 16, David. So they're all adult.
One is a single family household, right?
Yeah, no, one would be where we only have data from a single adult in that household.
But is that telling you what the household size is or not?
Not from that single variable. We would have to then look. So other participants may have not given consent to have their bloods collected during the nurse visit, or
okay, so this is not telling us household size. This is just telling us the number of individuals in a household that we have data.
We have this data for. Yeah, but we can obviously we have that data about the household composition that we can use when we come to analyze our biomarkers.
Okay, great. Thank you.
Yeah, great. So I was just going to go through those different biomarkers. So I've mentioned the biomarkers that we have from the blood. Some of them are not directly from the blood. Some of them are the other sorts of biomarkers that you might be familiar with, things to do with body fat, grip strength, lung function. And then the genetic and the epigenetic, we have numbers, again, you can see here, we have around the epigenetic data of around 3,650 participants. And in the households there, we've got couple, again, we don't know from this presentation of the data whether these are married couples, but we can find that out when we use the other variables to do with household composition. But we have households here where the epigenetic data is there is 628, 650 over the two waves. So we're talking about 1,200 odd households where there are at least two participants who've given blood that we have analyzed for the epigenetics. And then there's there are upwards of 60, just over 60 households where we have data for 3 participants in a given household. But the genetics is much higher. We have the genetic profiles of over 10,000 people. Well, on those numbers, considerably more. Right, next slide. And this is the proteomic data. So this is the most recent of the new omics that Understanding Society has presented as a data asset available for analysis. And this, we have 6,180 adults. And essentially the proteomic data is primarily to give us information about cardiometabolic and neurological processes. And the cardiometabolic ones, I think, are going to be very useful when it comes to understanding the health consequences of living at or below or in the composition of our social baseline. So, right, now, after that whistle-stop tour of the data assets and a little bit of an understanding of how the households are, the information we have at the household level, if we return to those papers that I very quickly introduced at the beginning, so the social allostasis paper and Jim and David's groups as organisms paper. If we look at this quote here from page 478, the social allostatic in the paper that Saxby published, that ultimately we propose the idea that social allostatic load as an organizing principle for understanding the impact of social regulatory systems of outside sources of stress, whatever those sources may be. In the past 2 decades, research on social regulation of brain, body, and behavior has increased substantially, and we are closer than ever to understanding that dyads and groups as emergent regulatory systems. So in our view, it's time to start thinking about not only about the internal regulatory functions of those systems, but also the larger context in which those social regulatory systems exist and how their context can facilitate or harm the fundamental regulatory functions of social relationships. So in my view, that gives us, I think we can frame a question using that as a starting point and leveraging the Understanding Society household as a functional union of biological analysis, where to take David and Jim's construction of understanding the household as an organism, and then using, depending on how we think we might best use either the proteomic or the epigenetic data, try and analyze the extent to which we can understand the social allostatic consequences of living in different sorts of functioning groups. So these 2 bottom quotes come from the paper that I mentioned, the groups as organisms paper that David and Jim published in the Clinical Psychology Review. And on page 6, again, because it's the baseline embedded in a social ecology, our predictively regulating brain perceives its own resources to include those as groups to which we belong. Our group's resources are our resources are our groups, or more likely, they're only the resources available to the dyad or the group. Critically, our challenges are the group's challenges, and our group's challenges are also our own, all of which point in a similar existence, just to the challenges. So this, when I was reading this the first time, this struck me that this very elusive term in social epidemiology and in sociology and in criminology and other related fields about the importance of neighborhood effects, and that actually perhaps we might be able to use the Understanding Society data, both the data that we have about the individual level, the household level, and the area level social information that we have about the participants, and then see the biological consequences of living in these different neighborhoods, in these different households for these individuals. And then finally, the final thing I've highlighted here was another quote from Jim and David's paper. If I just concentrate on that final sentence, more than that, SBT, social baseline theory, suggests that the group level model of the self is the default, that our baseline estimates of resources availability assumes embeddedness in a social system, treating departure from social systems as inherently threatening and requiring obligatory stress response. So it was actually that particular, I'm just going to focus on that for a second. It was that particular quote from that particular paper that really triggered my thinking on this subject. I think that we could test that using the data that I was already working with at Understanding Society, with Understanding Society. So the protein C-reactive protein is a marker, very well established marker for physical health and chronic exposures to stress. It's a marker of inflammation. So I proposed and actually sent an email in the first instance to David, which was that we could compare participants living alone with participants living in households with others, adjusting for both the nature of their relationships and the relationships quality, the usual demographic, health, socioeconomic factors, group membership beyond the home, their genetic and epigenetic confounders, and explore the consequences of departing from our expected level of social embeddedness on our stress profile. So it was at that point that the process of trying to get a group together to examine some of these initial ideas that I'd had motivated by these papers from David and Jim and Blair, to see whether we could leverage the Understanding Society data asset to test some of these hypotheses, some of these research questions. And there's some other questions that have occurred to me and Blair subsequently that we could also use the same, use the Understanding Society to examine. So household epigenetic clocks. I won't go into what epigenetic clocks are now, but we can do in questions if you'd like. Looking at the biological and chronological ages of households and understanding the social gradient of the healthy life expectancy at the household level instead of the individual level, which is traditionally where it's been analysed. I also wondered to myself, what variables would one use to compute the measure of social allostatic load? And what sort of, would one use a measure of relationship quality or individual well-being as an outcome if one was looking at social allostatic load? And then I also think an area that I'm particularly interested in is try to understand neighborhood effects through this lens. So using the social information that we have on each of the individuals, the household, and the area that they live, understanding each of those different geographies as a potential unit of analysis, and with the different constellation of factors in the composition of a derived variable to try and drill down into what the neighborhood effects are that impact on people's health. And then there's just a couple of open ideas. So the biomarker user guide, if one puts in glucose, so basically our energy packs, it yields the glycated hemoglobin marker. So how could we use this biomarker? How could this biomarker be leveraged to test social baseline theory? And in the proteomic guide, if one searches energy, there are a number of other different proteomic markers that we might use to also examine energy in that data asset. So I'm sort of, actually, Anna's here, and actually I'm now going to just talk very briefly about one of the papers that Anna published last year. So on household epigenetic clocks, an idea. So Anna and Mina, with some other colleagues from, no longer colleagues of mine, but when I was a doctoral student, I never met Wen, but I know Yachin. So Anna and Mina recently published this paper using a theoretical frame of the marital resource model and stress model. And the marital resource model proposes an increase in health-enhancing resources such as economic, psychosocial, and social support is associated with partnerships. And the stress model puts more emphasis on relationship disruption, positing that divorce and widowhood may cause deleterious effects and short-term stress and sustain chronic strains, which undermine people's health. But when I looked at this paper and read this paper, I could see the work that they'd done through this lens of social baseline theory. Now, the findings from the paper were ones that we're already familiar with. So they'd used this DNA methylation process to basically calculate people's biological age compared to their chronological age. And we have a standing understanding that people in relationships tend to are healthier in older age than people who are widowed or divorced, or people who'd remained single throughout their lives. But Anna and Mina demonstrated this, but also found that there's a positive age acceleration in people who divorce, and which is expected. But interestingly, some of the other findings were that men at an early age who were not in relationships had a slower biological age than their contemporaries who were married, so that they had less, well, as it were, bioenergetic burden evidenced in their DNA methylation, whereas men who were older, who were single, they had accelerated age compared to their contemporaries. So once the men had not had the responsibility of a young family, but did have the burden of being individual into old age, there was a different profile to their chronological, so their chronological age was now, their biological age was in advance of their chronological age. So these illustrations demonstrate the importance of partnerships in the aging process, in particular marital change through divorce and widowhood for positive age acceleration in adults. So that's just a quick illustration of work that was done by Anna and Mina. But there's also ideas coming from within the social baseline theory group. So Becky's and Spara, in their social baseline state of the science New Directions review from a couple of years ago, have a series of open questions that look a little, well, particularly the first one, are not unlike the questions, and the second one is much more one of the questions that I'm interested in addressing too. So, can the concept of social anesthetic load be applied to dyads and groups to produce new hypotheses and discoveries? Well, we think yes, and that we think understanding society is one way of testing that. And to what extent is the impact of social context on perception and the physical world? And that's my particular focus, because I'm very interested in the physical environment and particularly the built environment. Okay, so the stage that we're at is, so this is still emerging work. We're still in the process of formulating these research questions so that we can make our formal application to Anna's successor and to Mina. So Anna was formerly the research officer responsible for these data assets at Understanding Society, but subsequently, now that she's part of the SOCB program herself, that post has moved to somebody else. But we're in the process of formulating our questions and then requesting the variables from both the social data and the particular biological variables that we'll want, so that we can test some of these hypotheses. And the way that we're going about formulating our research questions is by using open science framework and working in collaboration with some of the people whose names we've been, whose work we've been looking at and whose names I've been repeating. So not only Blair, but Jim, Aaron Davies, who's at Oxford, and Lane Beckers, Rick, who as David said, he's my atavistic grandfather through Rick O'Gorman, and then Darby Saxby, who's the person who led on the social allostatic load paper. So we're formulating our theories and ideas here before we make the application. And why have we chosen the open science framework to do that? Well, Open Science Framework is an open source web application that facilitates open scientific practices. And by providing this online platform, the Open Science Framework aims to make scientific research workflow more transparent. We're very keen on making sure that, first of all, if there are other collaborators that want to join us, that they will have access to all the materials and all of the work to date, but also to facilitate that collaboration and to allow for evaluation and reproducibility of our published findings. So many journals and funders are now requiring, or at least incentivizing, people to use the Open Science Framework to manage their research process. So that's the end of this short presentation about where we're at. So thank you for your time, and Blair and I are keen to answer your questions.
Awesome. Well, let's get back to our gallery view.
Yeah.
So such an amazing set of possibilities. Liano, you're quick on the mark there. Go ahead.
If you wanted to go talk for a little bit, I've got a couple of. This was really great. I really enjoyed it. I've got a couple of quick things and one maybe larger one. First of all, from a biological perspective, we are obligate socialists because we are no longer biologically equipped to survive in the wild on our own. We need a community providing additional services and so on to allow us to survive. I was wondering if there's a way to make a strong evolutionary connection from that to this assumption about that we assume that we're in a group. And then the other thing is, I'm wondering do we have a baseline assumption about what size our group is, and or does that vary with culture? And does the perceived size of the group change the effect of the effect of these effects?
Thanks, Leon. I think I'll let Blair answer that first, and then maybe I can contribute once Blair's made a. You okay, Blair? You're on
muted,
mute,
Blair.
Okay, sorry, I'm on my phone and I'm not quite sure how to mute or unmute here. I'm used to doing this on my computer. Right. So okay. So I think that your so I'm sorry. So you had three questions, it sounded like. And so I'm trying to remember how to formulate your first question.
The first
So you're
question was we're obligate socialists because we are not
Right.
biologically equipped to survive in the wild on our own.
That's
So
what social baseline theory is saying. So I wouldn't say that we're biologically unequipped. I would say that we are actually, what we are doing is we are trained to the costs and the gains of acting in our environment. And so it's not that we're biologically unequipped, it's that we're sensitive to the presence of others because that changes the cost of acting in the environment. Okay, so that would be the first one. And then the second one you asked about, I'm sorry, I should have been writing that down. I was too busy trying to figure out how to unmute. So your second question was,
do we have an assumed baseline of what our group size is?
yes, I think that's a really great question. I think that is, so in my work, I talk about individual differences and I talk about how this is dynamic. We don't know. I would say that from my perspective, I have not come across anything that would say that we would have a certain group size, because there's also a lot of research that suggests that it's not necessarily group size, it's about quality versus quantity. And so there's a little bit of research into whether it's better to have a broader, shallower network versus a deeper, closer network. But I think perceived support is more important than any other variable that I've been able to find. Although again, we haven't really dove really deep into this, but from my perspective, it would be perceived support from the perspective of the person, the individual that you are studying in that sense. And so there's not a group number. And then the last one you asked about, like, does this differ culturally? I think was your.
Yeah. Well, yeah. Is there maybe the assumed size that we take on is one driven by our culture rather than anything innate?
No, I wouldn't go that far. So again, what a group. So while we're all social, not all groups are equal. And so I don't think you can assume a certain group size or a group presence of a group or absence of a group. Because again, it also, that's the point that I like to highlight, is that it depends on individual differences in your experiences. For some individuals, the presence of a group is a cost. And so that would not necessarily be a benefit. And so I think that it's a little bit more complicated than that.
Okay.
Yeah, that actually, segues to my set of questions. That so much is contextual, and that was represented in the Jim Cohen study that you cited, in which you had the main effect of holding hands, alone, stranger, and so on. But then you had those correlations with the quality of the relationship.
Yeah.
And I guess one of my questions is, can we get that kind of context from the longitudinal study? And you said that perceived support is one of the strongest, and that makes sense because then the individual is factoring a lot in their estimation of perceived support. I can well imagine, for example, and just to pick the example of Norway, that because the state provides so much support, then individuals often decide to remain single because they don't need economic support from a partner, and they find it easier to remain single. They still have relationships, but they don't have a marriage relationship, and so that gets really complicated. So how do you? There'll still be main effects, just as there was in Jim's study. So I think crudely, you're going to get effects from these basic categories, but how much can you delve into the context of all these things?
That's a really interesting question. And the idea that cohabitation versus, well, that's really interesting. Well, so I'm really hopeful. I think that might be a question more for Anna or David. How much can we dive into this?
So yeah.
But my hope is that is exactly the kind of question that we could tease apart. I do know, I think there's some research that suggests that in terms of your question specifically, that people who actually have a ceremony, maybe not necessarily legally married, but ceremonies, tend to correlate with sort of higher levels of commitment, et cetera. But yeah, I hopeful. So I'm not sure exactly which variables we might have. That's a question for Anna and David.
Yeah.
But that's exactly the kind of question that I would love that I think that this data set offers for us.
Okay.
So
Oh.
yeah, so there's modules in which, in the social data side of it, David, where we delve into relationships, not just with the spouse, but also relationships outside of the household as well. So we'll get an idea not just of the relationship quality with the significant other, but also their level of engagement.
Right. Okay.
In social groups outside the household.
Yeah. Okay. Well, let's go to Chip, and then I'll come back in.
I want to follow up maybe on David's notion of context, and I have absolutely no idea how I would answer the question I'm about to ask, and that's a different version of context. So I'm by training a survey researcher, political scientist, and the notion of having a longitudinal panel survey of a gazillion people where you were also able to do things like draw blood is just remarkable. And I've encountered the British study in context of Raj Chetty's work here in the States, which is methodologically very different, but lets you also do these kinds of things. But I couldn't not think of the communities the two of you work in. Middlesbrough, which is, to say the least, a disadvantaged part of the UK, having lived in a town that looked down on Henley, which is exactly the opposite part of the UK. So in that case, how would class and the like enter there? And for you, Blair, working in Lynchburg, where you've got your place, which is a pretty secular school right next door. Sure, physically, I mean, I live 100 miles from you, but having the community of Randolph next door to Liberty University, how does context come into play in the kinds of things that you're seeing? And again, remember, I'm a political scientist and peace builder. All the stuff that you're talking about is really quite removed from me, but I'm just fascinated by both what you can do with this study and with the kinds of questions that you're asking.
Yeah. David, did you want to chat about the built environment? I feel like first, and then.
Yeah. Oh, I think Chip's question about the groups, those broader groups, the social class, working, the way that people identify whether. So you mentioned Middlesbrough. I know that your question isn't specifically about Middlesbrough, Chip, but the data that we have, we know we can link the data with the postcode, so the zip codes of the participants, so that we all know which region they're from, and we'll also know from other data, if we choose to use it, that the amount to which they identify as living in a neighborhood that they resonate with, the extent to which they feel that they're part of their community, and then what that community is that they feel that they're part of, whether it's to do with it might be a sport, or it might be some other social aspect that they're part of. And I think this gets to one of the things I'm excited about is David and Jim's work, notion that all of those groups have a different value to us, and they all provide different resource to us at different points and for different purposes. And obviously some of those groups come with a cost, as Blair was talking about. Some relationships come with a negative energetic consequence. But generally we try and associate in groups where the net kind of global benefit over the term of the relationship is a positive one. So I was thinking about that when you were talking, that you're right, but we do have enough of the social information to give us a better idea of the context, instead of it just being at the household or just being in the immediate neighborhood of the focal participant that we have the data for. So I don't know if that kind of, I think it starts to answer your question, Chip.
From my perspective, I think David said just what I would say, which is that to begin with, so from my perspective where I am, it is really interesting because I actually reside right outside of Charlottesville and I work in Lynchburg. I think that's one of the interesting aspects of this data set, because then you can start to begin to build models where you can ask the questions about not just where you live, but if there are changes in circumstances over time, you could model that. And I think, to me, honestly, that might give you a lot of a more predictive value or information about where people feel or how their environments are affecting them. If you think about something along the lines of upward mobility or stagnation or things along those lines, to me, would be the most interesting kind of thing because from my perspective, this is about, from a Bayesian perspective, updating priors and expectations. And so I think that changes from a baseline are going to affect you the most negatively if you're talking about like sort of going down and that sort of perspective. Did that answer your question? Did I not? I hope I answered your question.
I mean, since I couldn't have answered the question, yeah, you did great. But before David remembers to unmute himself, one of the cool things about British postal codes rather than American zip codes is a lot more about the person from the postal code than you do from, I mean, my zip code includes extremely wealthy neighborhoods like the one we live in and quite poor neighborhoods. That's much less common in the UK.
Yeah, again, one of the reasons why this data set is so exciting, because I think it's just so rich with a lot more information than we would typically see. So
what's the difference between a British postal code and the American zip code?
It has 6-letter number combination in it, so it can. I think our postal code when we lived in the UK was like our street only.
Yeah.
It can sometimes be down to the odds and evens on a given street.
So it's a much finer grain.
Yes.
That's wonderful. That's really great. So the longitudinal aspect, I mean, this is a longitudinal studies, but so far we haven't talked much about like analyzing the same families from T2 compared to T1.
Yeah.
So speak about the longitudinal database.
Well, not one problem, but the fact is that the biological data was only collected over 2 waves back in 2010 and 11, 2009 and 10. So the longitudinal.
Oh no, he froze.
Longitudinal aspect isn't something that we could. Am I back in now?
Yeah. We lost about a paragraph.
Yeah, okay, sorry. So, yeah, I do know that there's, in fact, Anna might know more about this than I do. Is there, I understand that there's going to be some more biomarkers collected, Anna, in later waves now. I don't want to put you on the spot, but.
Yeah, there will be.
Yeah. So we will be able to start to possibly ask questions like that, David, when we get the next round of biomarkers, and then we can look back over a decade to see how things have changed with those biomarkers, but they're not collected as regularly as the social information is collected, which is on this annual basis.
Yeah, well, I would think that there'd be some kind of study where you could, first of all, establish correlations between biomarkers and other information, such as survey information, which you do have more of.
Yeah.
And then after establishing those correlations, then you can proceed with the larger database, if that makes sense.
Yeah.
Anna, do you know how broad that the next round of biomarker data collection is going to be?
I'm not too sure on the details, sorry.
Yeah, no problem.
So let's talk a little bit about the group that you want to form to help to frame hypotheses and so on. So, that's because I find that very exciting. I'd like to. And then another thing is, what kind of new studies? I'll ask two questions at once. First of all, forming a group around this. And the role a pro-social world can play in that, because we have a sizable research community now, and then the new studies that could be done that are informed by the longitudinal study, because I, from the very beginning, I thought, at the very least, in a new study, we could use the same biomarkers that are being used in the longitudinal study. But there's, I think, much more is possible about starting new research on the basis of the analysis of this. So I'd love to hear both of you on that, on those topics.
So, well, I'm probably the wrong person to ask about what kind of group we want to start, because I'm more of the everyone and everything genre. So, but I think that what, and David, jump in at any point and correct me if I say anything that you might want to correct. But I think from my perspective, there's just so many possibilities that we were sort of trying to collect, and I wouldn't necessarily categorize, but maybe I would say categorize sort of various avenues of research that you could take social baseline and sort of run. And so that's reflected on the Open Science page. And so from my perspective, it would be, who wants to join and come up with ideas and hypotheses and commit to projects in that way, with mostly, from my perspective here, all of these ideas, and I want to do it all, which is not possible. But so sort of what avenues are you most interested in, and where would you like to commit your time and your bandwidth and your own resources? So for the groups, that's that perspective. I have from my own dissertation, I did a lot of glucose studies, which I haven't sort of dabbled with in a while after my dissertation. So my argument was that my argument is one that I make, which I actually don't provide a lot of evidence for in my review papers, but I did collect evidence for in my dissertation. I've yet to publish these studies, which is that particularly in the case of visual perception, social support literally signals physiological resources. And that's actually a quite complex argument. And so from my perspective, I'm most excited about this possibility of kind of opening that line of research back up and saying, hey, we have all of these wonderful glucose markers and different things along those lines, that we could explore some of these claims about, if what we're really saying is that we are, social resources are reducing the metabolic burden, I think the kinds of data points that can address that question are available in this data set. And that to me is the most exciting aspect of it. But also David has some really great ideas about looking at built environments and connecting that with visual perceptions. I'll let him talk about that. But I'm really excited about his ideas as well. And I think they're just absolutely fabulous. And I can't wait to sort of start collaborating on those in a little more detail.
Have you found, if I could inject, have you found that the open science framework is useful for these kind of conversations?
Define useful. I think that from my perspective, I appreciate the open science framework because it provides unlimited storage space, and also it provides a timestamp. And so the kinds of questions we're about to do with this data set, I think it's important to sort of get down on paper that, hey, here's what we're thinking, here's our ideas, here's our hypotheses, and it provides a timestamp before you go sort of wading into the data, because I think that'll be helpful in sort of saying, hey, here's a confirmatory hypothesis and here's an exploratory hypothesis, and you can move from there. So I think that it's very helpful in that perspective. As a wiki page, I think it depends on how much is the group willing to put in the time to use the page. To me, it's sort of like any other, and I hate to say social media, but it's not social media, but it is. So the platform is only as useful as we are willing to put the work into it to make it so. And so from that perspective, I think it would depend on who's collaborating. If you really weren't like super, if people weren't, really big kind of fans, then I think you can use it and integrate it as much or as little as you want. You could use it just as a repository for data and ideas, and collaborate mostly online and other platforms and venues, or vice versa.
Yeah. Okay, great. David, I kind of butted in with that question.
No. So I was thinking that I'm still really interested in how one would construct a social allostatic measure using the data that we have on the individual level, from the individual level biomarkers. So I think that's a good question to have. We've got one of the working groups is about how would one constitute a measure of that using the markers available to us, the proteomic and the epigenetic markers available to us through the data asset. So that's something I'd like to focus on. But as Blair's mentioned, and as I alluded to in my brief introduction, I'm really interested in the extent to which the built environment plays a part. So actually one of the motivating studies for my work, David, is your study from community perception back in 2011. And I've been trying to synthesize your work with Jim on groups as organisms with the built environment as a bioenergetic resource, and it's understanding that environments where maintenance is poor through that lens of Dennis Proffitt's economy of action framework, right, being in those places solicits a predicted energetic expenditure just to get by, just to get through, just to get around things. And living in those environments is a tax in itself, is an energetic tax. So yeah. One of the studies that I've done that I didn't show us today, because I didn't think it wasn't where the conversation went or where the seminar went, was that I demonstrated using understanding society that if you live in unkept gardens and where the built environment is dilapidated, you have a higher rate of inflammation, controlling for all of the usual other social factors, so education and socioeconomic position, and your BMI, all of the other factors controlled for. The physical environment has this big impact on your levels of inflammation and your physical health. So I'm going to the extent that I can use the information that's in the data, in the survey, as well as the biomarkers, to drill down into understanding exactly what it is about the physical environment that acts as a index, acts as a crystallized consequence of the social environment, is what I'm going to be focusing on.
That's a study you just cited that's already been done?
Yeah, so it's under review at the Human Ethology, but it was actually very happy to share it with you, David.
Yeah, I brought up a couple of slides from my early Binghamton work that you just cited, that one. Let me just show a couple. And the point of showing it, I think, is in part just to stress how micro this is, how microspatial this is, that when you do a map of prosociality here with dark neighborhoods is our neighborhoods in which most of the students are highly prosocial and light is low prosocial. On a scale from 1 to 100 for an individual, neighborhoods can vary from 1 to 50. That's how rugged the prosocial landscape is. And this is even more micro. I mean, this is just such a tiny street. We call this the valley of distrust, that on one end here you get, and this is very much the built environment. This is kind of nice suburban homes. You go down here, mostly rentals. This is 1 minute on your bicycle that you do this. And then this study here, where we were contacted by the principal of this elementary school, who said 90% of the problems in my school are caused by about 50 students. And these are second graders that are acting like hardened teenage, high schoolers in their insubordination, and they're in first and second grade. And so we said, well, give us their street addresses. And all of them, without exception, fell into one of these prosocial valleys. Coloration is reversed here. And so, my God, I mean, and this is just like a 3-minute walk from one to the other. And I think that the result you just cited, David, is going to be found. I mean, there's probably going to be massive effects when you relate the biomarkers for people here versus people here 3 minutes away. But you better get the grain right, because if you don't, then you're going to be lumping heterogeneity. And so it'd be nice just to keep that in mind. And a question I have is, how fast does this take place? Because in prosocial, all the time, we're forming people into groups, and we're making those groups highly prosocial. And the individuals respond, of course, in terms of what we're measuring. But could we expect a biomarker response, like in glucose and some of these other things, in a period of weeks or even days? I mean, it could be like very fast, I suppose. What should we expect in terms of the speed of response? And also the context that if you provide a really great group within a single context, but you don't change the rest of their lives, then what does that do?
I think those are some of the questions that we need to answer. That's a really good one. How fast does it change? I mean, I would imagine, well, first, I think it's going to depend on the biomarker that you're using, because some of the biomarkers are more stable over time, and by definition, some of the glucose markers, that is what they are. They're stable measures over time. What I did in my dissertation, which again isn't published, although I did replicate the study and the finding, was I found that hand holding helped. So I had participants fast when they came into the lab, and they at one point were holding hand. They either sat alone, or they held hands, or they held hands with a friend. And this was mediated by attachment style, which I saw the chat earlier about attachment style, and that is absolutely 100% correct. This is mediated by attachment style, but essentially what it found is that for the people that were securely attached, holding hands with a friend helped prevent a further drop of glucose in their bloodstream. And so I think it depends on the level and the marker that you're looking at. I think that the problem is it's very hard to isolate and see these kind of short-term variables in the moment. So for example, these kinds of things like rises and falls in blood glucose, you see a lot of different sort of various studies about replicating and not replicating. And I think it's because there's a lot that goes into regulating blood glucose. And so we're looking at a very small piece of the pie in terms of, variance explained there, but an important one. And so that is that question about how fast does your baseline change, you know, I don't know, and I would love to find out.
Yeah. Well, Liana, you have, we're pretty well close to time here, so let's Liana, and then I might just ask for a quick checkout for people, which we like to do. So go ahead, Liana. Oop, you're muted, I think.
Sorry about that. I was just gonna say what that all was it seems very much related to the example study we talked about last week, where there was two groups of children, one of which was given a bunch of aid, and the other one was not, and then then there was and then they had opposite to expected responses, where the people that were given aid but then it was cut off got worse responses than the people that had never gotten the aid. And I just, it seems to me that this kind of that looking at that kind of those kinds of social interventions through this method would give us an indication of what the actual causal relationship is there. And it seems like this is a good theory that matches very well with what she was talking about last week, and I cannot remember the name of what
Barbara Oakley. Barbara Oakley.
Right.
Yeah. No. Yeah, that sounds as to what something I would predict, that giving aid might raise your baseline and withdrawing it suddenly would drop you below your baseline.
Exactly.
Yeah.
Exactly.
Okay.
That was my prediction.
Well, let's have a quick checkout. If I could ask people who could ask everyone, but Lauren, we want to just check out with a reflection, and then Hal, and then.
Yes, definitely. Thank you so much, David and Blair, for that beautiful presentation. And Blair, sorry about the Zoom troubles.
I'm gonna assume it's all on my end.
You did a wonderful job, even from your iPhone, so I appreciate that. And I really love this presentation. It really reminds me of the importance of others in being social and having others in your life. And throughout in my mind has been wandering to the implications that all your work and your research questions and your studies will have for, I guess, prevention science and helping to make the world a better place. So I appreciate it.
Great, thank you. How about you, Hal?
All right, well, it's very interesting. It's a very nice addition to the knowledge commons. And I have one thought, which is I wonder if a variable you've ever looked at is, if you ask people, how much do you value facts? I think there's some people, if you ask them that, they would say their attitude would be, what have facts ever done for me?
And how do you think that will relate to some of these biomarkers and stuff like that, Hal?
I don't know about relating to biomarkers. I think it may well relate to how people function in the world, if various aspects of that are being measured as well.
Mm-hmm. Right.
How do they vote, maybe?
Yeah, okay, great. Chip, how about you? You can bring us out. You can be the, I don't know if Carolina and Maria are present and would like to say something, but otherwise, Chip, you can be the one that brings us out.
Well, then two quick points. First, you brought out the social science nerd in me. I was involved in the late 60s and early 70s in at least three studies, one in France, one in the States, one in the UK, where we did at least 3-wave panel surveys of voters. And in the UK, it was a British national election study. And it was just very interesting to come back to that 50 years later. But the second thing that's really intriguing to me is that I've never thought about how you would incorporate the kind of biological data that you all are able to gather that certainly would not have been technically feasible in my day, and would not be technically feasible in US public opinion polling, or probably UK public opinion polling right now, because no one goes to the people's homes anymore. It's done by phone. And I can't imagine how much money this must be costing the citizens of the UK to do this project. And we should thank them. And given what I do for a living, I need to think about the pro-social aspects of all of this for the work that I do, which today was spent thinking about what we can do about Gaza, and then talking to a woman who runs the Youth Peace and Security Program for the United Nations Staff College. And again, all of these folks need to be thinking about these things. So that was long-winded, so I'll shut up.
Yeah, I'm sorry, I don't mean to detain us too long, but I was just made a connection that I don't want to, I'll want to make sure that it has, which is dual inheritance theory, which is a big topic for us and for generalized Darwinism. And when I talk about it, I'm always presenting a graph which, first of all, rehearses, your genotype. Everyone has one of them, and influences just about everything that could be measured about you, your phenotype. Then you have your symbotype, that's your symbolic meaning system, also influences just about everything that can be measured in you, the very same phenotype. And the symbotype and the genotype interact with each other during your lifetime, in addition to co-evolving for our entire history as a species. But if we focus on the symbotype genotype interaction, or more generally, the whole biological system, not just the genotype, but the whole biological system, as in some kind of very tight dialectic with the way we think and see and view the world, then the whole biomarker part of what we've been talking about is one of the things you'd really love to know and relate not just to your physical environment, but to your symbotype, the particular worldview that you have. And it's there where Hal's comment that, what about the kind of person who says, what have facts done for me? Or is in some ways a very alienated person. That's that worldview, which might be very different than the environment that they inhabit. It could be so the biomarker symbotype relationship, or basically studying this in the context of dual inheritance theory, is something that, and then I can't seem to stop, but Steve Hayes' most recent work is he's on a real crusade that research has to be idionomic. In other words, every individual is enough of a separate complex system that you must gather longitudinal data on individuals before you aggregate. And so he is now setting up the capacity to do that kind of research, in which the client is in, like, providing information. It's like the experience sampling method kind of, and one wonders what kind of biomarkers could be incorporated into that kind of study. It would have to be something which is pretty easy to gather, not too invasive to gather. And it's there where I think that the idea of relating this to new research, the point I made earlier, is like super important. What kind of biomarker measures should you be employing? Which ones are the easiest to employ? Which, in kind of the budget version, what would you be able to do? And then what might you do if you had more funds, and so on. So anyhow, that's all what I hope transpires as we develop this relationship.
Yeah.
Okay, so thank you once again, and thank you, David. Thank you for staying up. Now, is it past midnight now, or?
It's 12:30. Yeah.
Yeah, okay, well, bless your heart for staying
Well,
up.
thanks to Anna and
I guess that goes
Carolina
to you
and
too,
Maria
Anna.
too.
Oh
Yeah.
my,
Yeah.
Yeah.
All right, good night, everybody.
All right, good night.
Right.
Thank you, everybody.
Bye-bye.
thank you.
Yeah.